this is your Chapter 3 lecture, Part 2. This is covering sections 3 and 4, which is covering specific type of proteins, enzymes, and the cell's ability to inhibit their actions. Okay, so a little refresher on what a potential energy diagram is. As you can see, on the y-axis we have our free energy, and then as the reaction takes place is on our x-axis. So a couple things I want you to see. One, I wish they kind of would have did this in last chapter, because they were talking about delta G, and we should talk about delta G here as well. But every reaction requires a certain amount of energy to start us off. Okay, and that energy, as you can see right here, is the activation energy. Okay, so the activation energy is the energy required to have a reaction start. Now, important right here, notice that delta G along the side over here, delta G is not affected by the activation energy, because as you reach this energy, or as you're going up the energy barrier from the reactants place right here, as you go up the and heading towards that transition state, well, that amount of energy is eventually going to be released again once you've reached the level of the reactants again. So even though you're putting energy in to get the process going, you're also going to be releasing that amount of energy. So it's not really we're not really paying attention to the gross amount of energy being produced, but more of the net amount of energy being produced. Okay, a couple notes here. You should see right here that delta G is going to be negative. Okay, can you imagine that dotted line being 0? The delta G would be less than 0, so it would be negative. And of course, this being an exergonic reaction because of that. If the products actually ended up being higher than the reactants, then that would be an inorganic reaction. Okay, so this graph right here is showing the importance of enzymes. Now, enzymes are a biological catalyst. They're usually composed of a protein and either a non-protein component. That component can either be a cofactor, which is mainly like ions, okay, like iron and zinc. But you'll also have what we call coenzymes, which are carbon-based components that are themselves not a part of the enzyme, but they are important for the enzyme to do its job. Okay, so looking at this graph, we should see two different lines. One's red, one's blue. Okay, now the blue line is representing an uncatalyzed reaction. Now, an uncatalyzed reaction means there was no catalyst, or in this case, because it's a biological system, there was no enzyme present in this process. So it took a lot of activation energy in order to get to the transition state, which is pretty much the top of the heap. And then once that heap rolls back down, and we are going from the transition down to the dashed line again, it's a lot more energy going in, and then we'll end up obviously with having a lot more energy coming out. But like I said, we're not paying attention to the gross, we're paying attention to the net. Now, if you look, delta G is going to be net either way, right? It's going to be net negative, whether we have the blue uncatalyzed reaction or we have the red catalyzed reaction, meaning that an enzyme was present in this process. Now, as far as what enzymes do in order to lower the activation energy, there's two things that must happen in order for any reaction in the body to occur. Now, obviously, this is happening between molecules. Now, if the reactants, okay, are in the right orientation, okay, so that's one, right orientation, and they have enough energy going into the process to have a reaction take place. So the amount of energy going in, so the activation energy, right, we need to have enough activation energy to make the reaction take place. Also, the two reactants, in this case, because let's say I'm doing an anabolic process in which I have two molecules and I'm combining them to become one molecule, right? Let's say two glucose molecules, they're going to come together to, or say a glucose and a fructose come together to make a sucrose molecule, right? Well, one, I need to have that glucose molecule and that fructose molecule to be in the correct orientation, meaning they need to be lined up in the right place, right? And they need to have hit each other hard enough to stick together. Okay, kind of what we call a collision theory. They need to not only run into each other with enough energy, but they also need to run into each other with the right orientation. Okay, so what an enzyme does is enzyme will, one, grab onto the two substrates. It'll grab onto the sucrose, or grab onto the glucose, it'll grab onto the fructose, and then it'll arrange them in the proper orientation, and then it allows, what happens is, because they're already forced in the right orientation, it doesn't take quite as much energy to make them come together. And that's what it means by lowering the activation energy of a reaction. Because what happens is those enzymes are putting the pieces in the right places. Okay? So here we have our sucrose molecule. Now in this case, I'm breaking it apart. All right? So for step one, we have, let's go over some anatomy of an enzyme, right? The enzyme is, like I said, a protein and a non-protein component together. Here, they're not distinguishing the two of them. But this enzyme is sucrase, all right? And the substrate, which is meaning is the substance that the enzyme is going to act on, is sucrose, which is, as we know, a is a combination of a glucose and a fructose molecule. Okay, so that disaccharide is going to fuse into the enzyme at the active sites. Okay, now those active sites are usually having, you know, those nonpolar hydrophobic interaction spots that will match up with the ones that are on the sucrose. So usually your active site on your enzyme have compatible either ionic charges or has a place for a hydrogen bond to form, and it will match up exactly with the substrate. That's what makes them so specific. Okay? They will only match up in certain places because they're only built for certain jobs. For example, sucrase is built to break down sucrose. It is not built to do anything else, and its active site will represent that. Okay, and as you can see, they're just using the shape, overall shape of the molecule, but it's not just shape of the molecule. You're also looking at shape of, or not shape, but the types of ions that are present, or the amino acids that are present on the inside of the active site, with their R groups being exposed. Right, those R groups are going to be very specific for parts on the sucrose. Okay, when the substrate and the enzyme bind together, they form what we call an enzyme substrate complex. This is where we're going to have it's kind of like the holding cell. It's the process. It's getting ready to happen, right? So now they are currently in the right orientation. So that we're pretty much holding this molecule still so we can bring in water and undergo a hydrolysis, right? Hydrolysis is splitting them up. We can bring in a water to undergo a hydrolysis reaction, and then the two products, the glucose and the fructose, are released, and the enzyme itself is unaffected. So one enzyme molecule can actually catalyze or break apart or act on many substrates. Okay? So as you can see right here, this is just a three-dimensional molecular picture of it. I know it's not really three-dimensional, it's, you know, but showing a 3D model. And there's an empty active site, and then we have the substrate showing in, and all the different parts of the substrate are going to be exposed to all these little lines, and all these little lines are essentially, you know, they could be, you know, places where hydrogen bonds can form, places where hydrophobic interactions can form, places where ionic attractions can form, which will grab on to the substrate. Okay. So if you look at the bubble, when the substrates bind to the active site, the two halves of the enzyme move together, changing the shape of the enzyme so catalysis can take place. So what it's talking about is what we call an induced fit. So when the substrate enters, it actually, if you look at the picture, the enzyme changes shape. Well, it's part of the chemistry that goes on inside the active site. All those interactions are occurring, and it's causing the enzyme to change shape, which will allow it to do its job more readily. So now that we know what an enzyme does, let's talk about the ways that our body can prevent our enzymes from working. Now you're thinking, well, why would we want that to happen? Well, understand, we don't want enzymes to just be made and then never stop. There's always going to be a purpose. We need to turn them off sometimes. And there's one way to turn off enzymes, and this is known as inhibition. Okay. Now there's two types. There is a kind of a permanent inhibition, which means there's going to be an irreversible process, or I think in the book they call it irreversible. It's an irreversible inhibition. Okay. The other one is obviously a reversible inhibition. This is an example of how a chemical known DIPF, okay, is going to pretty much make acetylcholine esterase, which is an enzyme important for the destruction of acetylcholine, which is a neurotransmitter, and we'll get to that later. But the DIPF is going to bond. Okay, so if you look down here in the picture, okay, the hydroxyl group is the the side chain of a serine, which is a amino acid, and that serine is found inside the active site. There's a hydroxyl group on the outside of the serine. Okay, well, the DIPF is going to come in and it's going to react with that hydroxyl group, and if you notice in the next picture over, okay, so now we're on the picture on the far right, the serine is now no longer bonded to an OH, but now it's just bonded to an O, and that O is bonded to the rest of the DIPF molecule. But even though off the top of my head I couldn't tell you what DIPF means, I know that that F is probably meaning fluorine, since there was a fluorine in that molecule, and now that fluorine is gone. Okay, you can also notice in the top picture, that's the little green ball. That little green ball is gone now. Chances are, when this happened, the H from the hydroxyl group and the fluorine from the DIPF reacted, and they became hydrogen fluoride and released. And that caused this ester linkage in between the serine and the rest of that DIPF molecule. Well, once that happened, that enzyme I've changed the structure of that enzyme. That hydroxyl group is no longer on the edge. Now I have a whole bunch of methyl groups, or CH3s, on the edge, which are not going to react with anything. So now I've practically, forever silenced and neutralized this enzyme. This is what we call an irreversible inhibitor. We stopped it and we cannot change it. So what will happen is usually the cell will, catalyze or catabolize this enzyme and then harvest all the amino acids to make another type of enzyme later. So the other type of inhibition is the reversible inhibition. And there's two types. We have competitive inhibition and we have non-competitive inhibition. So with the competitive, all that means is that there is another substance that is going to compete with the substrate, and it's going to fit in the active site just like the substrate does. So if the competitive inhibitor is being produced, that means we want to silence the enzyme, right? So for example, let's look at say sucrase, right? Well, what if we want a certain amount of sucrose in our blood, but we have a lot of enzyme there? Well, if we have the enzyme, the sucrose is going to be broken down by the sucrase, the enzyme. Okay? So if we have a competitive inhibitor come in, it'll block the sucrase from breaking down the sucrose, and that way we'll have we can maintain a level of sucrose in our bloodstream, or a level of sucrose in our cells. Okay? Now the non-competitive inhibitor is similar to a competitive inhibitor, but it just doesn't, as far as it will prevent the activation of the enzyme. But what it does is it will bind on the bottom, or bind on some other part of the enzyme, and in doing so, changing the active site. And when it changes the active site, the substrate will not be able to bind to it. Okay? And of course, both of these can be reversed. That's why they're called reversible inhibitors. Okay? So just to kind of show a couple different ways how enzymes are regulated, okay? On the right side of the screen, we'll have an activator substance that comes in and activates and opens up the active site. So just like when we did here, this is to turn off the enzyme. Well, sometimes we have the enzyme there, but we need to turn it on in order to get it to work. And that's what activators do. And also, the idea of protein kinases, I'm going to kind of skip that. It's just I can't really do anything in phosphorylation. I can't really do anything with this side yet, so I'm going to mainly focus on the right side. Okay? So the activator comes in, turns on the active site. The active site can then attach the substrate, and then the substrate can be turned into a product with the active enzyme. Okay? So there's ways to activate enzymes and there's ways to shut them up. Okay? So this is what we call feedback inhibition. So sometimes the product, the end product of our enzyme action is going to end up turning off the enzyme to begin with. So for example, here I am at therione. This is the starting material, right? This is an amino acid, right? And then what will happen is there's an enzyme on this big green arrow is representing an enzyme that's going to convert this amino acid into alpha ketobutyrate, okay, which is an intermediate product. And then these next arrows, these next 4 arrows, are a series of enzymes that are going to eventually turn it into isoleucine, which is our end product. Well, here's the thing. Isoleucine, if there's enough of it, will actually come over here to the big green arrow and turn off the first enzyme. And if that first enzyme is turned off, then you can't make more isoleucine. So it's kind of, this is the whole idea of negative feedback, or a feedback, is the product is what's going to control whether or not I need to make more of it. All right? If I don't have enough isoleucine, then there's nothing over here to stop this big green arrow enzyme turning the amino acid into the intermediate, and then I can continue on with my other processes to make more isoleucine. All right? These graphs right here are just going to show, well, hopefully we can figure these ones out, right? The blue and the red and the purple are representing 3 different enzymes. And then if we have pH on the bottom, this is showing what we call an optimal pH. Enzymes like specific pH levels, okay? Pepsin obviously likes a little more acidic, and makes sense. It's found in our stomach to break down proteins, okay? Chymotrypsin is found in our intestine, which is much more neutral. So when we get to the digestive system, we'll talk more in detail about how pepsin is neutralized and how chymotrypsin will then take over in the protein digestion. Okay, so this is just showing that enzymes like certain pHs. Right? And this one is showing what? Reaction rate versus temperature. So obviously, there is an optimal temperature. Enzymes like certain temperatures. If you go too high, you can actually denature the protein and it'll destroy it. Or if you go too low, then it's just the molecules aren't moving fast enough to begin with to reach the enzyme, so the substrates really can't come together, or the substrates really can't be broken apart because there's not enough thermal energy in the system to get things going. Okay? If you have any questions, make sure you ask in class.